There Might Be Hope, Chapter 5, An Unloved Flower. For Dust Bowl survivors, plowing came to equal erosion. Erosion came to equal dust storms, and dust storms meant hunger and scarcity. Just like Adam and Job, though, they plowed for a reason, so do we. Among other reasons, plowing eliminated unwanted plants from competing for sunlight, nutrients, water, and space with the food they wanted to grow. We call those unwanted plants weeds. Plowing rips apart any existing plant root systems and buries them alive under the soil, giving the young seedlings we plant a head start. To feed the world's rapidly growing population, weeds had to be plowed under, but to save our soils, we had to find a way to farm without plowing. Solving this age-old agricultural problem wasn't how the world's best-known herbicide came into being, though. In 1942, in an effort to preemptively answer the legitimate threat of biological warfare, the U.S. Secretary of War selected George Merrick to direct research on technology that might be utilized in biological warfare. In modern echoes of the Romans salting Carthage, one promising theory was to destroy Axis food crops by spraying them with herbicides from planes. Among nearly 1,100 chemicals being experimented with on crops at Camp Detrick in Maryland, two drew considerable interest. Recently synthesized 2,4-D and its cousin, 2,4,5-T, showed promise. According to Merrick in a 1946 letter, it was only the rapid ending of the war that prevented field trials in an active theater of synthetic agents that would, without injury to human or animal life, affect the growing crops and make them useless. In 1945, 2,4-D made its entrance into commercial agriculture, and as agricultural historian Gail Peterson notes, that the public's enthusiasm for the newly unveiled herbicide was equaled by their universal ignorance on the chemical and how it worked. Such an enthusiastic reception prompted more chemical companies to begin producing their own 2,4-D herbicide formulations. In only 2 short years, 30 unique 2,4-D-based herbicides were available, and over 5 million pounds had been sold to American farmers. Since the 1950s, about 30 million pounds of 2,4-D have steadily been applied to American agricultural crops every year. Because pesticide sales have historically been both loosely reported and underreported, the figures are certainly on the low side. By the mid-1960s, production had grown to 10 times that, and 2,4-D was found in 6,000 different formulations. Said by some to have initiated the agricultural revolution, 2,4-D's success at obliterating weeds in pursuit of untainted crops ensured its rapid ascent to becoming one of the most widely used pesticides in the world. Weeds decrease the yield and lower the quality of crops in gardens, on farms, on the plantation, and on the range. Weeds disfigure the land, affect the national health and the national pocketbook. Death to Weeds. Dow Chemical Company, Death to Weeds, 1947. George Merrick didn't live to see the mobilization of the work he began with 2,4-D in World War II, but 2,4-D and 2,4,5-T would ultimately realize their original purposes. In the end, they were sent not to annihilate field crops in the European theater, but to defoliate the jungles of Vietnam. Roughly 20 million gallons of rainbow agents accomplished their mission, eliminating enemy cover and clearing the way for U.S. base camps. Developed for war, but designed for agriculture, the rainbow agents came in 7 different formulas. Agent Blue was based on arsenic, but Agents Green, Purple, Pink, White, Orange, and Super Orange were all based on either forms of 2,4-D and 2,4,5-T, singularly or in combination. Like DDT, the D in 2,4-D stands for dichloro, or 2 chlorine atoms. Its primary lethal agent. 2,4,5-Ts, or 2,4,5-trichloro, phenoxyacetic acid, also relies on chlorine as its lethal agent. The mechanisms by which chlorine causes death differs between herbicides and insecticides, but the result is the same. Often the particular potency of these formulas can be attributed in part to their structural similarity to essential plant growth hormones. These herbicidal Trojan horses fool the plant into welcoming them as essential hormones, and that is precisely what they do. As with the insecticides, though, from 2,4-D's introduction in 1945 through the 1970s, weed resistance grew. So we began mixing herbicide cocktails. Layering different formulations of chlorine-based pesticides together increased their individual efficacy. By 1970, over 100 different herbicides were in general use. As with the insecticides, most were halogen-based, thus compounding our bioaccumulation problem. A solution to growing weed resistance and poisonous halogen accumulation in the environment had to be found. Once more, Monsanto led the way. In 1974, they brought a novel idea to the table, simply called glyphosate. Commercially, it would be most frequently dubbed Roundup. The unnatural union of two naturally abundant and essential nutrients, glyphosate, is disarmingly simple. It is the combination of glycine and phosphate, both essential nutrients to plants, animals, and people. Glycine is a neurotransmitter, a serotonin producer, an essential DNA building block. It supports mammalian mental health, sleep, memory, and the digestion of fatty acids. It also regulates the body's immune system and heals inflammation. Phosphate is also essential. Bones, teeth, and cellular membranes all rely heavily upon phosphate, and without it, muscles won't contract. Nerves won't function. How do two such brilliant and vital ingredients combine to form a deadly herbicide? In plants, one of phosphate's jobs is to build a multitude of amino acids, including the essential amino acids tryptophan and phenylalanine. This amino acid building process is known as the Shikimate pathway. It's glyphosate's job to stop it. The Shikimate pathway is tricked into accepting glyphosate as simple phosphate, but with the addition of glycine, it's much bigger. Like an assembly line where one slightly wrong part clogs the entire line, glyphosate settles into phosphate's normal space and halts any further production line progress. Essential amino acids tryptophan and phenylalanine can't be made, and the plant starves to death for lack of these essential amino acids. The enzyme that would have been produced by the Shikimate pathway is called EPSP, and it is essential for the synthesis of aromatic amino acids and almost all other aromatic compounds in algae, bacteria, and fungi. Glyphosate isn't lethal because it's poisonous. It's lethal because it creates starvation through nutrient deficiency. Glyphosate is considered to be the active ingredient in Roundup and is labeled as such. But in recent years, Roundup and other herbicides, inactive and inert ingredients, have been receiving increasing attention. It is actually those inactive ingredients that comprise the bulk of herbicides today, and because they are considered to be inactive, companies do not have to disclose these ingredients. These inactive ingredients mix and marry with the active pesticide and water to help them slip smoothly through the sprayer and stick to the plant's foliage. They're also designed to enhance absorption and persist upon the leaves of the plant. Because they have been considered to be both benign and proprietary, chemical companies have not been required to divulge these ingredients. Until recently, no one considered testing these ingredients for safety. New research, though, is showing that these inactive ingredients are often far more lethal than the actual active toxin. Heavy metals such as arsenic, lead, nickel, and petroleum products often comprise the bulk of these inactive carrier agents. One such petroleum project, POEA, is considered to be up to 2,000 times more toxic than glyphosate itself. Farm fields and farm soils are literally being coated over and over again with variations of petroleum and heavy metals. Despite all of this, because a new generation of plants sprouts up every year, glyphosate resistance, like insect resistance, was inevitable. Knowing this, scientists wasted no time in beginning to develop the next solution, and as with insecticide halogens, the immediate answer was to apply more glyphosate. From 1945 until 1996, the amount of herbicides that could be applied to a field were dictated by the crop's life cycle. Herbicides generally had to be applied before planting or after harvest. Spraying during the growing season might kill crops. With agrobacterium in their pockets, though, scientists needed only to reach through the genetic portal to engineer a crop that could survive without tryptophan or phenylalanine. They could do what had never been done before: spray growing crops with herbicides. The first genetically altered crops that could survive without the shikimate pathway were named Roundup-ready crops. Later, as patents for similar crops proliferated, these genetically modified seeds would become generically known as GMOs. For the first time in human history, seeds were patented. If the wind carried seeds or pollen with patented seed DNA from a farmer's field to his neighbor's field, and the plant grew, farmers could and were often sued for theft by the seed patent owners. In fact, some 410 farmers and 56 small businesses across 27 states were sued by Monsanto for patent infringement from genetic drift and or reusing seed from a previous crop, which was forbidden. Many farmers lost the lawsuit and then their farms. In the 1980s, we barely understood mycorrhizae and the soil microbiome better than Roman farmers, but soil compaction had already been connected with diminishing yields. Reasonable and rational fears of a second Dust Bowl loomed large as the best of America's farmland poured into the Mississippi and emptied into the Gulf of Mexico. The answer to the plow, the answer to Leptis magna, the answer to the Dust Bowl, the answer to the Great Plains washing down the Mississippi, it was all wrapped up in glyphosate and GMOs. They had to work. Our future depended upon it. We couldn't keep plowing like we had been. We knew we couldn't keep pouring halogens and arsenic into our farm soils. By genetically engineering crops to withstand the toxicities of pesticides, farmers wouldn't have to till anymore to eliminate the weeds that fought their crops for limited sunlight, water, and nutrients. The method was simply titled no-till farming. Although human history repeats itself frequently, the title of being the first generation in all of human history to bravely attempt no-till farming on a commercial level surely belongs to us. Fields planted with Roundup-ready crops could not only be sprayed with glyphosate before and after the growing season, as they had been in 2 previous decades, they could now also be sprayed 2 to 4 times during the growing season without killing the GM crop. Predictably, sales went through the roof, and to a point, farmers increasingly adopted it. Why wouldn't they? No-till farming offers tremendous savings on fuel and labor costs. According to U.S. government estimates, A farmer working a 1,000-acre field can save around $10,000 in fuel and 70 hours of work a year by switching to no-till. That's a nice vacation to Bora Bora in the dead of a frozen Iowa winter. So why are only about 20% of U.S. farmers using no-till 40-ish years later? Perhaps many farmers have realized that conventional no-till came with fine print. Some call them superweeds, others call them frankenweeds, ragweed, milk thistle, cat's claw sensitive briar, lamb's quarter. Not only are these plants not deterred by herbicides, they seem to thrive in spite of them. 2, 4, or even 6 times in a single season, glyphosate and other agri-cocktails rained down on our farm soils. The more we sprayed, the more weeds grew. Impervious to chemical assault and seemingly wed to the embattled and dying soil microbiome, these unexpected nuisances could not be deterred. Eventual resistance had been anticipated, but not with this speed and tenacity. No one plants them in the middle of crops, yet they emerge and thrive. Between resistance to insecticides and resistance to herbicides, we are at a dead end. Or are we? Beneath the crop dusters, beneath the tractors, beneath the sprayers, soaked in insecticides, herbicides, and inert ingredients, lies what remains of our embattled soil microbiomes. It is here that superweeds love to make their homes. These nuisances to our preferred food crops love poor and dying soils, and they are here to rescue them. Superweeds, indeed. These superweeds tend to be plants that naturally pull lots of nitrates, a form of nitrogen, into their leaves. By doing so, they help to rebalance and restore a healthy carbon to nitrogen ratio in farm soils, and thereby begin to prepare the soil for soil microbiome restoration. When they die, their stalks, stems, and leaves will provide food to help add even more carbon to the soil for microbes to feed upon. Some of these weeds also pull heavy metals out of the soil. There is an undeniable beauty to these weeds. In spite of our determination to annihilate them, they are here to save us. They are here to fight for us. They are here to begin detoxifying our soils. If the superweeds aren't actually nuisances, but heaven-sent heroes, are the insects really as bad as we have made them out to be? There is a group of agricultural scientists who believe that all plant and parasite interactions are nutritional in nature. This small contingent espouses that insect invasions are not bizarre, inexplicable, paranormal heralds of apocalyptic devastation to be battled with all our resources. Insect invasions prior to the 20th century generally followed periods of intense cultivation and extended drought. With hindsight, we can see that crop insect invasions served a larger, beautiful, and ultimately essential purpose. Like vultures and sharks, these insect invasions were the undertakers for nutrient-starved and dying plants. We weren't aware of how unhealthy the plants were, but the insects knew. The theory of trophobiosis espouses that insects only attack plants when the plant's biochemical state meets the nutritional need of the parasite in question. In other words, insects can intuitively tell when a plant is healthy and strong. These plants are unattractive to potential predators. But when a plant is nutritionally weakened, insects do what they're designed to do: consume the plant and recycle nutrients back into the soil microbiome to help ensure its health for future plants. When these insects die, their own bodies will further help rebuild the soil microbiome as it's on its path to complex nutritional recovery. The presence of insects en masse ought to be a warning alarm for us that our soil microbiome is failing and our plants are starving for nutrition. Our trouble is not that insects are doing their job in trying to rebalance a failing microbiome. Our trouble is that our crops are weakened and sick from generations of living on IV drip nutrition. By means of their inert and inactive ingredients, pesticides, whether insecticides or herbicides, are designed to stick to plant leaves. Plants, like soils, breathe, and inevitably, some pesticide is inhaled into the plant. Some may also be absorbed into the soil and plant root systems. Whether through leaf, stem, or root, plants absorb pesticides. Their response: first shock, then, like any trauma victim, demonstrable panic. All measurable growth and protein production halts as plants shift into red alert detoxification mode, trying to eliminate the poison. Plant cells begin breaking down the complex proteins and carbohydrates of their own tissues back into simpler amino acids, nitrogens, and sugars. Sugars begin pooling in the extremities, and tissues begin to swell within the circulatory system. In humans, we call this edema, and as with people, it's primarily found in plant extremities or the tips of the leaves. This pesticide-induced edema is not visible to the naked eye, but it's scientifically measurable. Simply because the plant didn't die or wilt does not mean it is unaffected. It just means that the quantity of toxin it ingested was insufficient to kill it. We've long held the absence of plant death to be indicative of plant health, but have we set the bar too low? This systemic shock from pesticide trauma temporarily increases the amount of simple sugars in the plant cells, making them simply irresistible for insects. Like the aroma of fresh bread or pastries wafting from a French bakery, plant stress beckons insects with the readiness of their favorite meal. We might as well be ringing the dinner bell with every pesticidal pass down the farm row. Insects are not drawn to a certain plant simply because it is a particular species. Insects don't have a favorite vegetable per se. They're drawn to particular crops in particular fields because the crops in the fields are growing in poison, sick, and dying microbiomes. These soil microbiomes lack the mycorrhizae, humic and fulvic acids, bacteria and protozoa, along with the complex proteins, nutrients, carbohydrates, and available minerals that would have grown strong, healthy, and nutrient-dense plants that would be less appealing to insects. In observable science, every reaction follows an action. It may have taken us a while to locate and measure the initial cause, but here we are now. Perhaps halogen gases, petroleum carriers, heavy metals, and genetic manipulation aren't necessary to prevent insects from devouring our crops. Perhaps we simply need to heal and restore our soil microbiomes. That's the end of Chapter 5. Thank you so much for listening. If you enjoyed this, please like and subscribe to my channel. That way you'll get notifications for the next chapter. If you're interested in learning more about behind-the-scenes research and just more in-depth information about this particular chapter, you can support me at Patreon. The link will be in the notes below. Thank you so much. Bye.